Good morning, everyone. My name is Kim McAllister, and I'm director of the Center for Neuroscience and a professor in the departments of Neurology and Neurobiology, Physiology, and Behavior. And I want to welcome you all to the second day of our virtual NeuroFest event. If you were here yesterday, we hope that you had a lot of fun, and we're really glad to see you again to be here to learn more. If you were here yesterday, though, I want you to know that I'm going to give the same introductory remarks as I did yesterday for the new attendees. It'll take about 10 minutes. So if you don't want to hear that again, go out and grab another cup of coffee. And if you do, it's nice to see you. So as many of you know, we had to cancel NeuroFest for the first time last year, right at the start of the pandemic. And what a year it's been. We hope that you and your loved ones have been able to stay healthy and safe during this turbulent time. And we hope to be back in person for NeuroFest next year. This is really one of my favorite events, and I'm excited to participate in this virtual NeuroFest event. Our truly awesome staff, students, and event co-chairs got creative and put together a really innovative program that some of you saw yesterday and that you're going to get to experience today. And we have a lot of fun in store for you. So I'm going to go ahead and share my slides. And what you'll see on this first slide is the schedule for today. So basically what we're going to do, I'm going to give you a little bit of a welcome, and then I'm going to tell you about neuroscience research at UC Davis and tell you about the Center for Neuroscience for those of you that don't know about us. Then I'm going to give some background on challenges in mental health research. Following my introductory remarks, you're going to hear two short talks from our excellent faculty, Dr. David Olson and Dr. John Gray. Then you're going to hear from our awesome students. 3 students are going to give a data blitz. And then after that, we hope that you will stick around and explore our interactive booths that our students have put together through the Gather platform. They're a lot of fun, and we're going to give you instructions during closing remarks about exactly how to navigate that. Okay, so we're all here today because we're fascinated with the brain and how it makes us human. Neuroscience is the science of understanding how the brain works. And this is the focus of research at the Center for Neuroscience on campus, where many of us work. Now, since many of you are actually new to this event, to NeuroFest, I thought we'd provide a short introduction to the Center for Neuroscience and neuroscience research in general through a short 4-minute video that I'll show next.
We're interested in understanding how the brain works for a number of reasons. And the first is really based on an innate curiosity that we all have about who we are as humans. Our brain development and our brain function allow us to do all of the amazing things that we do, to learn, to make mistakes, to adapt, to plan for the future, to love, to interact. It really is about who we are as humans.
Figuring out the mysteries of the brain is one of the grand challenges of science. Ultimately, if we're going to solve this grand challenge, we need to bring together people from many disciplines. We need the biologists, we need computer scientists who know how to handle big data. We need statisticians who know how to infer patterns from these data. We need physicists and computational modelers who know how to integrate across all of these different brain areas and across the different scales from molecular to systems to cognitive to put together a holistic picture of how brain function is occurring.
Well, the brain's important because it is one of the most amazing and complex phenomenon in the known universe. We have 100 billion living cells that are kind of organized in these networks, all wired up together, and they're connected to one another through some trillion of connections. And somehow or another, through their coordinated biological activity, we're able to see, I can see you, I can hear you, and I can think my thoughts and have my feelings and communicate with your brains. And that is one of the great miracles we need to try to understand.
The Center for Neuroscience is a very exciting place. It's an interdisciplinary center here at UC Davis that brings together faculty from many different colleges and schools and many different departments. We cover all aspects of neuroscience research, from basic to translational, from molecular to cellular to systems to cognitive neuroscience. It's an exciting place because each faculty member brings together a team of neuroscientists within their lab that have specific backgrounds and perspectives on how to study how the brain works. And when we bring these together from all of these differing backgrounds, we have sparks of new discovery and new ideas that you don't have if you're housed in separate places. We still only know the very tip of the iceberg about how the brain works. Brain disease is incredibly impactful to individuals that have it because many brain diseases prevent us from being able to do what we do as humans, from being able to function in the world the way that we do. And some brain diseases are even more devastating because they start to impact our identities and our ability to interact with our loved ones. So we want to come up with new cures and ways to prevent disease. There are numerous brain diseases that affect almost every family, and we have very few cures or treatments. And here at the Center for Neuroscience, we are trying to understand how the brain works and goes wrong in disease so that we can come up with better treatments and ways of diagnosing brain disease much earlier. So the future of neuroscience is all about team science. It's about innovation. It's about speeding up discovery. It's about taking discoveries and turning it into impact. It's about improving lives.
Center for Neuroscience is the future of neuroscience.
So you can see that the research that we do at UC Davis is really broad, and we typically choose just one area to focus on during NeuroFest. As you know, this year's topic is mental health, which itself is a broad topic. Mental health is essential for us to function on a daily basis, and we take it for granted unless and until we experience disruptions to our mental health. And these disruptions can come in many forms. They can show up as ADHD, as autism, schizophrenia, anxiety disorders, depression, substance use disorders, or PTSD, just to name a few. And honestly, mental health is at the top of everyone's mind these days. The rates of all mental health disorders have dramatically risen during the isolation and stress that's been brought on by the COVID pandemic. And those rates were already too high even before the pandemic. The statistics that you see here are pre-COVID. Psychiatric illnesses affect 15 to 20% of the population worldwide, with 1% experiencing schizophrenia, almost 7% major depressive disorder, about 3% bipolar disorder, and more than 4% ADHD. And neurodevelopmental disorders also have a very high prevalence. Autism affects 1 in 54 children and 1 in 34 boys, making it more common than pediatric cancer, diabetes, and AIDS combined. So as if it's not enough to have so many people's lives impacted by neurodevelopmental and psychiatric disorders, there is an even bigger ripple effect of mental illness that's shown here in this graphic taken from the NAMI site, the National Alliance on Mental Illness. So people with major mental illness don't just struggle with mental health, but they also have an increased risk for other chronic diseases like diabetes or cancer. And it doesn't just affect the person with the illness, the families are also affected. Caregivers spend an average of 32 hours per week providing unpaid care for their loved ones. And it affects our communities. Look at these statistics. 37% of people that are incarcerated in state and federal prison have a diagnosed mental condition. And remarkably, 70% of youth in the juvenile justice system have at least one mental health condition. And depression and anxiety disorders cost the global economy $1 trillion each year in lost productivity. So imagine what we could do if we could come up with better ways to diagnose and treat mental health illnesses. We could help not only the people with these diseases, but really, really have a dramatic impact on our community and on our world. So despite the prevalence and the devastating impact of psychiatric disorders, there remains a pervasive stigma that prevents us from talking about these disorders openly, and more important, it prevents people from getting help. It can take years for people to seek treatment, if they seek treatment at all. This needs to change. We need to stand up against the stigma. We believe that there can be no health without mental health. And why is it that there is such stigma around mental health disorders when there's little stigma to other diseases? Why do we approach our mental health differently than the health of our bodies? It would be silly to think that we could improve heart disease just by telling ourselves that it's not really a real disease and we should just be stronger and power through our heart disease. So why do we do that when we experience issues with our mental health? It's important to realize that mental health is brain health. So we have to better understand brain health in order to develop new treatments to improve it. And just like for heart disease, brain disorders are caused in large part by things that we cannot control, like genetic variants or mutations, but with different genes involved for brain disease than for heart disease. And so it turns out that many psychiatric illnesses actually start early in development, even though the symptoms appear later. And those changes in genes that cause the disease often converge on changes in the number of connections or synapses that are made in the brain. So if we plot the number of synapses throughout the lifespan, what we see is that after birth, there's a dramatic increase in the number of synapses during infancy and childhood, reaching a peak around age 9. Then the synaptic connections are refined. Some synapses are strengthened, others are eliminated. And that happens during adolescence and probably into the late 20s, after which synapse number remains stable. Now, in schizophrenia, one of the most enduring and reproducible findings is a decrease in the number of synapses that are found across multiple brain regions. And conversely, some recent evidence suggests that autism, which obviously occurs at a much earlier age of onset, may be associated with an increase in the overall number of synapses found across multiple brain regions. And those changes in synapse numbers also come with changes in the information that's transferred across those synaptic connections. That information transfer is mediated by chemicals called neurotransmitters, and those neurotransmitters are packaged into these little spheres. When information or electricity is trafficked down one cell, it causes those chemicals to be released and bind to receptors on a recipient cell, allowing for information transfer. And you probably know the names of some of these neurochemicals or neurotransmitters, like dopamine or serotonin. And we do have drugs that target these neurotransmitters, like SSRIs, Prozac, for instance, that are used for depression. And these can dramatically improve people's lives. But many people don't respond to currently available drugs. And so we're still trying to understand how to better fix imbalances in these neurotransmitters, and even better, how to target treatments to specific brain circuits within the brain. So today you're going to hear about 2 projects that target different neurotransmitter receptors to try to develop novel therapeutics to better understand what causes the range of psychiatric illnesses that we've talked about. Now, a shared goal for many of us at UC Davis is to develop more effective diagnostic tools and treatments to improve the lives of so many people that are suffering from changes to their mental health. And we can't achieve that goal without discovery-driven neuroscience research or basic research. And the way that discovery-driven research is central to developing new treatments is shown on this last slide that I'll show you. So what we do is we identify the disease that we're interested in, and then we identify a cause for that disease, say for instance, a genetic mutation. And then we develop disease models, animal models, or computational models like you heard about yesterday that model that cause of the disease. And then once we've caused the disease in our model, we can look to see what the full range of effects of the disease are in altering brain development and function. Once we know what's going wrong in the brain, then we can develop new targets that we can then identify new therapeutics to fix what's going wrong in the brain, eventually testing that in the disease model to see if it rescues the disease-like behaviors. And then eventually we want to run clinical trials back in humans to see whether that novel therapeutic will actually work in human populations. So I hope that that gives you a little bit of a better appreciation for how important it is that we talk about mental health issues, that we develop more and better treatments, and that the central role that basic research actually has in achieving these goals. Okay, so now it's time to turn to the keynote talks for today's session. Before I introduce the first speaker, I'd like to encourage you to use the Q&A function on Zoom to ask the speakers questions. You can put those questions in at any time, and we will, in the Q&A session, we'll go to some of those questions. The speakers are going to try to stay on to answer some of those questions. So if you don't get them answered, don't get frustrated, we'll get to you and we'll follow up with that. If you have technical issues, please use the chat function and let us know. We're happy to help. Okay, so with that, I'd like to introduce our first speaker. Dr. David Olson is an assistant professor of chemistry and biochemistry and molecular medicine. He joined UC Davis in 2015, where he established a research program at the interface of chemistry and neuroscience. He coined the term psychoplastogen to describe small molecules that are capable of promoting neuroplasticity. Dr. Olson is also the president and chief scientific officer of Dysfunction Therapeutics Incorporated, a company focused on developing non-hallucinogenic analogs of psychedelics for treating various brain disorders. So the title of his talk is From Deadheads to Depression Meds, How Psychedelics Are Impacting Modern Medicine. Dr. Olson.
Thanks, Kim. And so my research group is really interested in developing new medicines for treating a variety of brain disorders, but we focus primarily on stress-related neuropsychiatric diseases, which include things like depression, addiction, and post-traumatic stress disorder, as well as neurodegenerative diseases like Alzheimer's disease and frontotemporal dementia. Now, a hallmark of all of these illnesses is really the atrophy of key neurons. And if you think of a neuron like a tree, the dendrites of the neuron would be like the branches, and the synapses, the actual points of communication between two neurons, would be the leaves. And so in these illnesses, essentially what happens is the leaves fall off and the branches get trimmed. And so we are not as interested in trying to treat the symptoms of these brain disorders, but we're really trying to cure them by regrowing those branches and regrowing those leaves. And so to do this, we've been looking for chemical stimuli that essentially act as miracle grow, regrowing dendrites and reestablishing synaptic connections. And so our search for these elusive chemicals led us in a pretty unexpected direction. It led us to start studying these classic psychedelic compounds, molecules like LSD, which is well known as part of the counterculture of the 1960s, psilocin, which comes from magic mushrooms, and MDMA, which is associated with dance party culture and rave culture. And these are all drugs of abuse, primarily due to their hallucinogenic effects, but also sometimes because of their stimulant properties. And so the first thing that we did was we took neurons grown in a dish and we treated them with these psychedelic compounds. And we found that they were really good at promoting the growth of dendrites and promoting the growth of synapses. And so these little orange protrusions here, these would be like the leaves on the tree. And of course, you can see that the branches on the tree on the left have grown substantially after treatment with the psychedelic compound LSD. Now, the issue with using psychedelics as therapeutics really has come down to their hallucinogenic effects. That is the main reason that they have abuse potential. And so our group was really interested in trying to use chemistry to engineer psychedelic compounds to lack a lot of the undesired properties, but retain their beneficial therapeutic effects. And so I'm going to tell you one story today about this molecule called ibogaine. It's a natural product that's found in a shrub in Africa. And ibogaine is pretty remarkable because a single dose of ibogaine can keep heroin addicts drug-free for up to 6 months. And a second dose of ibogaine can keep them drug-free for up to 3 years. And so you might be asking yourself, well, if ibogaine is so great, why aren't we using this to treat the opioid epidemic? And the answer is ibogaine isn't that great. There are a lot of issues with it. One issue is that it's really hard to produce in large enough quantities for the clinical trials that we would need. Another is its physical properties are pretty bad. It's kind of like oil in water. So it's not very soluble in water. And that's a problem because the vast majority of our body is comprised of water. And so drugs have to be soluble in water in order to reach their targets. And of course, the major issues with ibogaine are really related to its hallucinogenic effects and its cardiotoxicity. Ibogaine binds to an ion channel in the heart that causes heart attacks, and several people have actually died during ibogaine treatment for opioid use disorder. And so I'm gonna summarize a lot of work and just say that we were able to solve this problem by essentially giving ibogaine a haircut. We chopped off parts of the molecule that were not important for its function, and we just tweaked things here and there. And we produced this compound that we call TBG, that is a safer version of ibogaine that still has therapeutic properties. And so just to show you that TBG is safer than ibogaine, the first question I always get asked is, how do you measure hallucinations in a rodent? And so I'm showing you a rodent behavioral test called the head twitch response. And so when you give a psychedelic compound to a rodent, they'll have this really rapid rotational movement of the head. If you blink your eye, you'll miss it. So pay attention, it happens right there. And we know that the potency in the rodent head twitch response assay correlates exceptionally well with human hallucinogenic potency. And so you don't have to be a chemist to appreciate the evolution of these compounds. So going from the molecule in blue, this is 5-methoxy-dimethyltryptamine, a very potent hallucinogen that induces a lot of these head twitches. And we can just chemically evolve it towards this green compound, TBG. And you can see that as we do that, we can drastically reduce the number of head twitches until there are none. And so TBG is a non-hallucinogenic compound. And this is really the essence of medicinal chemistry: make small structural changes in the molecule to improve their properties. So in addition to being non-hallucinogenic, TBG also has reduced cardiotoxicity compared to ibogaine. So what I'm showing on the right are immobilized zebrafish, where you can actually see the heartbeat. And this is the control fish, and below it is an ibogaine treated fish. And you can see how much slower the heartbeat is. And so if you measure the ratio of the atrium to ventricle beats per minute, you can get an indication of how likely a drug is going to induce a heart attack. And you can see that ibogaine is very likely to induce a heart attack, but our compound looks very similar to the control condition. Of course, we wanted to make sure that in addition to being safer than ibogaine, that TBG still produces the same beneficial therapeutic effects. And for this experiment, we used special mice that basically have glow-in-the-dark neurons. And we used a specialized microscope to peer inside their brains. And then we can measure the number of essentially leaves. These little protrusions are sites of those synapses, remember, like the leaves on a tree. And so we can measure those before and after administration of the drug and see how many new leaves are sprouted. And what we find is that TBG increases the density of those leaves, those synapses, to a comparable extent as this hallucinogenic compound DOI. So next we wanted to see if TBG also has some beneficial behavioral effects. And so for this experiment, what we do is we give mice access to a bottle of water and a bottle of alcohol, kind of on a chronic intermittent basis. And much like a UC Davis undergraduate, after a while, the mice start to prefer the alcohol. And then we give the drug. And what you'll see here is that every single animal in this experiment, about 20 in total, had a reduction in their alcohol consumption after administration of TBG. And finally, we wanted to see if TBG would be useful for treating heroin addiction. And so for these experiments, we train rats to press a lever to receive an infusion of heroin. And we administered TBG at 3 different time points. And what we find is that no matter when we administer the drug, it has this protective effect on heroin relapse. And so all of these really incredible discoveries made at UC Davis, we're trying to actually use them to improve the lives of patients. And so I co-founded a company called Psychedelics Therapeutics that are trying to take molecules like TBG into the clinic for treating disorders like depression and addiction. And with that, I'd like to thank a few people. First, I'd like to thank you for paying your taxes, because the NIH funded all of the work that I described to you today. And of course, I really need to thank all of the graduate students involved in this. They're really dedicated, hardworking individuals who are super bright, and none of this would be possible without their efforts. Thank you for your attention.
So terrific, David. That was an awesome talk. We already have one question in the Q&A. I'd like to encourage the attendees, if you have questions for David, type them into the Q&A, and I'll do my best to get to those. I'm going to start with one of the pre-submitted questions that was submitted during registration. So, David, how do you propose that we change the public's opinion on psychedelics in order to support its integration into medicine?
I think that we need education. I think we need to, teach everyone what is really true about psychedelics and what is not. And so a common misconception related to these drugs is that they are addictive. And that is not the case. They're not like molecules like cocaine or heroin. If you give them to animals, animals will not self-administer these drugs. And so that is a very common misconception that people have about psychedelics, and they think they're dangerous because they're addictive. That's not the reason that they're dangerous. They are dangerous for other reasons. They do cause pretty drastic perceptual changes. And so really they can only be administered safely under the care of a doctor in a hospital setting where you can be closely monitored. But I think education is the number one way that we can change public perception.
Terrific. And you're doing a little bit of that today, right? Okay, so there's a question in the Q&A from Edwin Lopez. The question is the growth of dendrites going to help if you cannot tell them where to go?
That's an excellent question. And so, selectivity is really important. You don't want to promote plasticity indiscriminately across the brain. If you promote neuronal growth and plasticity in the so-called addiction circuitry, that's what molecules like cocaine do. I just told you that psychedelics don't do that. And that's because they promote neuronal growth in a different part of the brain, primarily in the prefrontal cortex. And that is because that is where their receptor, their target that they bind to, that's where it exists. And as a result, we are able to achieve a little more selectivity for the specific neural circuits that we activate in response to the drug.
Awesome. Okay, so the next question is from Claire Warshar. Dr. Olson, are you familiar with valerian and its possible heart effects or other side effects?
I'm not. I'm sorry. I can't say that I am familiar.
Okay. Claire, if you want to tell us a little bit about that in chat, I think people might be interested in hearing about it. We've got another one that was from the pre-selected questions, and this is a big one. I think a lot of us are interested in. So will we be able to prevent mental illness onset in the future?
I really hope so. I love the idea of using small molecules as prophylactics. And what I will say is there are, the research in this area is really in its infancy. And, the NIH is really primarily focused on trying to treat brain disorders, but trying to prevent them, I think, is a really important goal. And we know that we can do this with non-drug treatments like exercise, mindfulness therapy. There's a lot of other ways to kind of stave off some of these conditions. But there is some really exciting research related to the molecule ketamine, suggesting that if you administer ketamine before stressful events, that you can actually prevent behaviors that are related to post-traumatic stress disorder in rodents. And so I'm hopeful that someday we will be able to prevent these diseases.
That's really remarkable. Another question from the Q&A from Benitha Dubridge. Do you have models to address the functionality of the new outgrowths?
We do. And actually, John Gray, who's going to be talking next, has helped us with some of these things. And so we can actually look at functional effects using electrophysiology, and John has collaborated with us on that. So hopefully he'll talk about that maybe.
Okay, great. Let's see. So Ed Hardiman has asked, is the neuronal growth promoted by TBG long lasting?
This is a question that we're trying to address right now and to see exactly how long the effects last. So something that's really interesting about ketamine. So ketamine has been approved by the FDA for treatment-resistant depression. Its effects only last about a week. Now in mice, ketamine promotes neuronal growth that lasts for about a week. Something like psilocybin is being investigated for depression as well in humans. And it seems from the early studies that the effects are much, much longer lasting than ketamine. Months to maybe even a half a year after a single administration. What's really exciting is that in mice, the effects on neuronal growth are also really long lasting, much longer than ketamine. And so there was a paper that just came out a couple of days ago that showed that psilocybin can increase formation of synapses that last for, at least a month in rodents.
Wow. Okay, another one. How effective is microdosing at reducing hallucinogenic effects of LSD, MDMA, psilocybin, et cetera?
So microdosing is kind of defined as taking sub-hallucinogenic doses of the drug on kind of this chronic intermittent basis. There's a lot of controversy about microdosing because there's been no placebo-controlled double-blind clinical trials showing that it's efficacious. And a paper actually just came out recently suggesting that the beneficial effects of microdosing really come from the placebo effect. And so we don't really know if it is as effective as a high dose of these drugs. Now, the other thing to keep in mind is that if you're constantly stimulating these receptors, that can sometimes have detrimental effects. And our group actually ran a microdosing study in rodents, and we found that the chronic dosing of these drugs actually caused neuronal retraction. And so, I'm very concerned about the effects of microdosing, and more research needs to be done.
Okay, good point. So there's a question from Stuart Teal that comes together with a question that was pre-submitted. So I'd like to ask, we've got people that are interested in new research related to Alzheimer's disease, research on how to prevent Alzheimer's disease. And Stuart Teal wants to know, have you tried this with Alzheimer's disease?
So we have. Kind of. We're at the very early stages of this. So we are very interested in using our non-hallucinogenic drugs to treat Alzheimer's disease for a variety of reasons. Number one, they turn on that cell growth and survival pathway, which will be hopefully super useful for neurodegeneration. The other thing that we're really interested in is the target of these drugs are also found on immune cells in the brain. And we think that they might actually reduce neuroinflammation, which we know is a huge part of Alzheimer's disease. And so this is really early research, but we already have a couple of grants to look into this. So keep an eye out for that in the future.
Okay, terrific. Let's see, we've got another one from Vanitha Dubridge. And Vanitha, thank you for your questions. I know you asked some awesome questions yesterday too. So David, how does your TBG molecule work in the context of other drugs like SSRIs?
That is an unanswered question right now. I suspect that it will not work as well. And I think that's when eventually these get to clinical trials, patients will probably have to have a period of time where they go off of their medication before they start taking this new medication. And the reason for that is that SSRIs can downregulate the target of TBG. And so if the target is not there, the drug is not likely to be effective.
Okay. And there's another question from Edwin Lopez. If there are drugs that can make connections, are you looking at any drugs to extend the life of those connections in the brain?
Yeah, there's a lot of potential ways to do this, but what we find is really fascinating about these drugs is that they seem to be on kind of a positive feedback network. And so when you stimulate this pathway, it will produce a molecule that continues to stimulate the pathway. And this will go on and on and on for a period of time. And so we actually think that a single administration of these drugs will lead to neuronal growth for quite a sustained period of time.
Okay, so that should help the effects of the therapy to last. Good. Okay, we've got a few more minutes. Margaret Hill has asked, are you doing any research regarding insomnia?
We are not.
Okay, maybe an idea for the future. And then there was another pre-submitted question, David, that's interesting. I'd like to know the answer to this too. So psychedelics are good for relieving depression symptoms. So is there science for their use for personal growth purposes, like improving happiness?
Yes, actually, I think some of the early work in the 90s done by Roland Griffiths at Johns Hopkins speaks to this. And so what Roland found is pretty remarkable. He was giving psilocybin to just healthy volunteers, and then he was measuring things like their happiness and their empathy and just like, how they felt about the world. And he did this not just by asking them how they felt about the world and how happy they were, but also by asking all of their neighbors, do you think your neighbor is a nicer person, essentially? And he did this over the course of months. And what he found is that a single administration of psilocybin would improve mood and happiness and empathy for at least 6 months.
Wow, well, I could think of some uses for that in our world right now. Anyway, David, thank you so much for an absolutely stimulating talk and discussion. We really appreciate it.
Thanks for having me.
Great. And thanks to the attendees for awesome questions. So now it's my pleasure to introduce our second speaker. Dr. John Gray is an assistant professor of neurology in the Center for Neuroscience. As a psychiatrist and a neuroscientist, he's interested in how dysfunctional synapses can lead to many psychiatric and neurological disorders, including schizophrenia, autism, and Alzheimer's disease. In particular, his lab studies the regulation of the NMDA-type glutamate receptors that play crucial roles in synapse function. Through his work, Dr. Gray is striving to develop disease-modifying therapies for complex neuropsychiatric disorders. The title of his talk, and Dr. Gray, if you want to share your slides, the title of his talk is Reversing Synapse Dysfunction in Neuropsychiatric Disease.
Hi, welcome everyone. You can see here this picture of me from last year before NeuroFest. I look a little different now. I vowed to not shave until I was able to give you this talk, so here we are. As Kim mentioned, I am a psychiatrist, and because of this, I've really seen firsthand the limitations of our current treatments. And because of this, I felt pulled back into the lab so that I could study the fundamental biology of synapse function, with the hope that a better understanding of how synapses work and what goes wrong with them in disease could translate into new treatments that people so desperately need. Now, the brain is a very complex organ. It controls all of our thoughts, perceptions, actions, everything that makes us us. You can think of the brain maybe like a big novel, like War and Peace. It can be broken down into chapters like the hippocampus, which is a major memory center of the brain, and down into individual sentences. Here are the individual neurons of the brain. You can see here firing their electrical impulses. But the fundamental functional unit of the brain is the synapse. It is the site of connection between two neurons. It's how one neuron communicates to the next neuron. In this analogy, perhaps it's the single word. Now, to take an even closer look at the synapse, this is a very simplified representation of a glutamate synapse. Many of you have heard of serotonin and dopamine, but the vast majority of synapses in the brain are actually glutamate synapses. So glutamate's released from the first neuron and interacts with receptors on the second neuron. And one of the key receptors that glutamate binds to is called the NMDA receptor. NMDA receptors play a very important role in neurobiology. They can direct synapses to either get bigger and stronger or to get smaller and weaker. And this is a process known as synaptic plasticity. And the synaptic plasticity is involved in forming the circuits of the brain and is thought to be the cellular basis of learning and memory. So when we learn something, we have synapses that get stronger. Because of this really central role that NMDA receptors play in brain function, they've been implicated in a whole host of neurological and psychiatric disorders throughout the lifespan, from neurodevelopmental disorders like autism to neurodegenerative diseases like Alzheimer's. So the goal of my research is to understand the detailed molecular mechanisms of how NMDA receptors work. Specifically, how can one single protein regulate both the strengthening and the weakening of synapses? These are like very opposite effects. How can one protein do this? And then to use that knowledge to selectively modify synapses that are abnormal in disease. For example, if synapses are too weak, can we make them stronger? If they are too strong, can we bring them back down to their normal size and strength? And so if we take PTSD, for example, in PTSD, during a traumatic experience, there's this massive wave of synaptic plasticity in the brain. The brain wants to remember everything that happens in the environment and associate it with that traumatic event. Now, you can see that this is actually a very beneficial event for survival. If an organism survives a traumatic experience, they want to remember everything about that experience so they can avoid it in the future. So from an evolutionary perspective, this is quite useful. But in PTSD, this can lead to intrusive memories, nightmares, hypervigilance, exaggerated startle, among many other disabling symptoms. And importantly, these symptoms can then be triggered by very innocuous environmental stimuli that were associated with the traumatic event in the brain through synaptic plasticity. For example, if someone develops PTSD after, say, being mugged at gunpoint next to a pizza parlor, they may spend the rest of their life with severe anxiety attacks every time they smell pepperoni. Now, that's just kind of a silly example with the pepperoni, but because usually these triggers are actually very difficult to identify. But the question is, can we do something to reverse this disabling synaptic plasticity? To do this, we need to take a closer look at the NMDA receptor. Here on the left is the protein structure, and on the right is a kind of a representative cartoon of the receptor. Now, NMDA receptors have a very unique property that makes them very different from almost all receptors in biology. And that is, in addition to needing glutamate, like I said, NMDA receptors are a glutamate receptor. They also require another compound called D-serine. And it's actually very surprising that we know that as important as NMDA receptors are, we know very little about how D-serine is regulated in the brain, or even why the NMDA receptor uses it. My lab recently found that unlike glutamate that's released from the first neuron through the vesicles like Kim talked about, crosses the synaptic cleft and binds to the NMDA receptor, D-serine is actually coming from the same neuron that the NMDA receptor is on, though we still don't know how it's released or how its levels are regulated. But despite this lack of understanding of the mechanisms of D-serine release, we developed a hypothesis that of what D-serine could be doing. And we think that it could be involved in regulating that direction of synaptic plasticity. Specifically, maybe high levels of D-serine could lead to the strengthening of synapses, and low levels of D-serine could lead to the weakening of synapses. So how would we go about determining this and measuring this? Here's Eden. She's an extraordinary graduate student in my lab. She's sitting here at her electrophysiology rig looking at a slice of mouse brain on the screen, though it's actually a very tiny slice that's underneath the microscope. And what Eden can do is she can send very tiny little glass tubes or pipettes down under the microscope and into the slice where she can attach them to individual neurons. Sometimes we change the DNA of those neurons and we can make them like glow, like David had talked about, glow green so we can see individual neurons. But you can attach these pipettes to the neurons. And when you do that, you can record the electrical activity of the neurons. So for example, we can look at the electrical responses shown here from individual synapses. And you can see that these synapses have some different strengths. Some of those responses are bigger than others. Or we can look at the firing of the entire neuron, firing its spikes like you saw in the video. So in the experiment that Eden did, she measured the average response of a set of synapses that she shows here in this bar graph. And the first 10 minutes, the -10 to 10-minute point, is what we call the baseline. That's the average synaptic response at the beginning of an experiment. This is what an average trace looks like. And then she applies an electrical stimulus to the slice. And we know from years of experience and many labs studying this, that you can apply different stimuli, sometimes to strengthen synapses and sometimes to weaken synapses. In this particular example, though, she picked a stimulus that made everything stay the same. So she applied the electrical stimulus, and you see it initially, there's a reduction of synaptic strength, but it comes back after 40 minutes or so back to the original level. And that's actually where it comes back to is what we're looking at, because we're talking about something that's the source of memory, and memory is something that persists. So we want to see the persistent changes of synapse strength. So then she used this system to test our hypothesis. And our hypothesis, again, is that high levels of D-serine might lead to synaptic strengthening. And that's exactly what she saw, is that after increasing the D-serine levels on the slice and applying the stimulation, she got stronger synapses. And if you had lower D-serine, so if you reduced the D-serine on the slice, we saw a weakening of synapses. So it fit exactly what our hypotheses were. So we were very excited about this. Maybe we can use modifying the D-serine levels in the brain to modify synaptic strength. And this is very exciting, but this is actually a very challenging proposition. This is a picture of a very thin slice of mouse brain, maybe a millimeter square, showing the vast number of synapses in the brain. Each one of these red dots is a synapse, a glutamate synapse specifically. The human brain contains upwards of 1 trillion synapses, and individual neurons can have maybe up to 10,000 on a single neuron. And we don't want to strengthen all of the synapses or weaken all of the synapses, but only a tiny subset of these synapses that are part of a disease-involved circuit. So how can we do this? And this is actually this idea of targeting and modulating specific brain circuits is actually a major focus of modern neuroscience, and is probably the future of treatment of many psychiatric disorders. And this is in contrast to a lot of our current treatments. All the current treatments are shown in blue here, and most of them are very global. Things like electroconvulsive therapy and even most of our medications are very global. They're hitting things all over the brain. Though we do have a few currently available approaches like transcranial magnetic stimulation and deep brain stimulation that are a little bit more targeted, but there are many more experimental approaches that are in development. Interestingly, though, psychotherapists have been activating individual circuits in the brain for decades, particularly in the cognitive and behavioral therapies. For example, in patients with PTSD, they can undergo a type of behavioral therapy known as exposure and systemic desensitization therapy, sometimes using virtual reality, like you see in this image, to activate the fear circuits. And while doing that, they have learned ways to develop control over those anxiety symptoms using various mindfulness and relaxation approaches to reduce the impact of those circuits on their life, to help control the anxiety symptoms that get triggered by these intrusive memories. But perhaps instead we could use a drug treatment, maybe targeting the NMDA receptor, immediately before these sessions. Then when you activate the circuit, the synapses in that circuit could be selectively weakened. And by doing that, maybe only one or a few sessions would be needed to eliminate the response entirely. So this is a large goal of the research that I do. And I think this approach could be used in many other diseases, from ruminations and depression to obsessive compulsive disorder, along with these targeted neuromodulation modalities, to one day we can reverse the synapse dysfunction that occurs in neuropsychiatric disorders. But to get there, we really need the basic neuroscience, the type that we do here at the Center for Neuroscience. And with that, I'd like to thank you for listening to me. And I, of course, like David, want to thank the talented people in the lab who are actually doing the experiments. Here you see John Wong, who published the paper on the D-serine coming from the postsynaptic neuron. He just recently received his Ph.D. And Eden Berrigan, who's did the experiments I showed, and she's at one of the booths this afternoon. So please go and talk to her there. And of course, I'd like to thank my funding, including funding from the National Institutes of Mental Health. Thank you.
So John, that was just terrific. Thank you so much for a really awesome talk. You already have a lot of questions in the Q&A. So we'll go ahead and just start with those. Lynn Weaver asks, Dr. Gray, what happens when the synapses are too strong?
Hi, Lynn. That's a great question. I think the answer can be different depending on where in the brain they're at. So when we think about synapses being strengthened, we're thinking about synapses that are strengthened during a learning experience. So in the PTSD, for example, they're being strengthened during the experience. And we're calling them too strong in this particular instance because they are causing disabling symptoms for the patient. This is really normal synaptic plasticity. We want to just bring it back down so that these circuits do not get activated by innocuous things in our environment. So it's not necessarily that the synapses are too strong. They're too strong because they are causing disability for the patient.
Okay, and Lynn has a second question. Is there a correlation between the number of synapses and their weakness or strength?
In some diseases, yes. I think for example, in schizophrenia, as Kim mentioned in her introduction, there isn't what we think is an over pruning of synapses during adolescence. This is why schizophrenia really has an onset in late adolescence, early adulthood. There's an over pruning of those synapses. And a lot of evidence has shown that before there's actually a loss of those synapses, there's a selective weakness of too many synapses, and they become weaker and weaker, and then they're eventually removed. So if synapses across the brain become progressively weaker, they will be eliminated, and then you'll have a change in the overall number of synapses. It's a great question. Thanks, Lynn.
Great. Okay, so the next one is from Ruth Elkins. I think like many that heard about this, she's really fascinated about your brain slices. And so she asks, how are these tiny slices of brain tissue kept moist and alive?
That is the challenge. That is the critical part. We have to remove it very quickly to keep them alive, keep them healthy. We put them in a solution that's very similar to the chemical composition of the solution that our brain is in, our cerebral spinal fluid, and we keep them in that solution. We provide nutrients, and we also provide oxygen. We actually basically bubble the slice with oxygen, so that oxygen gets dissolved into the tissue. We dissolve this oxygen in the solution, and then when they're on the microscope, it's constantly being perfused with fresh solution that entire time. So it's a challenge, and we can keep them alive for the slices alive for up to 4 to 6 hours, and then that's kind of the end of our day. So we try to keep them healthy and alive as long as we can.
Yeah, so for the reasons that Dr. Gray just pointed out, studying neurons, and especially when they're alive, is incredibly difficult. And so, the students that we show recording from these, they have a lot of training, and they're really, really good at what they do. And we're very lucky to have the opportunity to do this, but it's a really challenging line of research. Okay. Dr. Gray, 2 more questions from Benita. So the first one is, where does D-serine come from? Is it from the diet? It is not the functional enantiomer in mammalian proteins. What is the molar, maybe composition?
Probably the concentration. Yeah, so D-serine is an amino acid, and like most amino acids, all of the proteins in our body are made out of amino acids. But interestingly, in biology, all of the amino acids that make our protein are actually called the L isoform. So these are basically just mirror images. And there's an enzyme that's only found in the brain. There's a little bit of it in the liver as well, but mostly found in the brain, called serine racemase. And this enzyme converts L-serine into D-serine. And this is actually how we've determined that the D-serine is coming from the neuron that the NMDA receptor is on, because that is where the enzyme is found that makes the D-serine. So we think L-serine is brought into the neuron and then converted locally to D-serine where it's released. And so it's actually at very low levels in the brain, but it was higher than was predicted because the mammals don't use D amino acids in general. So when it was originally found that the mammalian brain had high levels of D-serine. This was very surprising. This was Solomon Snyder's work back in the early 90s. And it was very surprising that there was all this D-serine in the brain. So then it was found that it was interacting with the NMDA receptor. So we do get a little bit from the diet. If you remove the enzyme that makes D-serine, you reduce the levels by 90 to 95%. So we actually do get some from our diet. Actually, I think most of it probably comes from bacteria in our gut rather than the food that we eat. But that's a small proportion of the overall. Most of it is actually generated in the brain by this enzyme that converts it from the L-serine. And its overall levels in the brain is, it's about 1/20th of what L-serine is. So it's very, very low relative to L-serine.
Okay,
and
terrific.
We think it's local.
Okay, so the second question is, how are NMDA receptors desensitized?
That's a great question, and it's a very interesting question that we know a lot about from what we call a biophysical perspective. So people studying NMDA receptors in isolation, we can see all of these desensitization processes. So desensitization is when a receptor gets activated, and it's activated for a period of time. Then it doesn't just turn off. It actually turns off, and it's turned off in a way that it cannot be activated again. This is the desensitization part of it. It's basically no longer able to be active for a while. And I think what's actually happening is people have found that when glutamate binds to the NMDA receptor, part of this desensitization actually reduces the affinity of the receptor for D-serine. So D-serine is no longer able to bind to the receptor as much. You need more of it around. So we think that there's a situation where when you have a lot of activity, a lot of glutamates being released binding to the receptor, that actually you need a bigger push of D-serine to actually overcome that desensitization of the receptor so that you can cause synaptic plasticity. So this is something that we're actually starting to look at using some low affinity antagonists, which are blockers of the receptor, to see if we can overcome that low level of binding. But we think that there's some interesting things that could be done with that, but we just don't know very much about what desensitization does in the brain.
Okay, great. We've got a few more minutes for questions, and I want to turn to a couple of more clinically related questions.
Sure.
So Claire is asking, has not schizophrenia been an assumed disorder for many past patients? It does not seem to have been based on physical evidence for some patients. And so I think what she's pointing out is the difference between the diagnostic criteria and the fact that you're showing that there are biological signatures in the brain. And I wonder if you could speculate on that and how biology might actually be used in the future for diagnosis.
Yeah, I mean, I think if I'm understanding the question right, I think this comes down to a lot of what Kim was talking about in her introduction, that as a public, we really perceive mental illness as something different from physical illness. And that's just not the case. The brain is an organ. All of our thoughts and our feelings all come from our brain. And these disorders that psychiatric and mental illnesses are disorders of the brain. So yes, historically and even today, people somehow put things like schizophrenia separate from other even neurologic disorders like Alzheimer's disease. But that's just not the case. Just because we don't see a specific clear accumulation of a protein like amyloid in Alzheimer's in schizophrenia doesn't mean that it's not rooted in the brain. It just means that the brain is very complex and we don't have a full understanding of it yet. And there are a lot of physical changes in the brain that we do recognize, including there's an enlargement of the ventricles in the brain. There's a reduction of synapses. This was originally found that there was a reduction in gray matter in the brain. And then people spent for decades counting the number of neurons in the brain and said, hey, everything seems about the same. The neuron levels are about the same. And it's only been more recently that we found that it's actually a loss of the synapses, like David showed with the neurons being like a tree, the branches get brought back and there's less leaves on the trees. So there are these very physical, clear structures. There's just nothing that we can easily use for diagnostic purposes right now. And this is something that we really need to be able to do better, is finding these biological signatures so that we can recognize and get clear diagnostic categories, which is a whole other issue, but very clear diagnoses so we can treat people with the best treatments for their specific disorder.
Right, and that's where we've heard two talks now that are really looking and focused on trying to identify new therapies. But as we identify those new therapies, they're also going to be identifying biomarkers that are signatures in the biology that correspond to the disease that we can then develop diagnostic tools against. And so we need more ways to diagnose the biological change in brain health that correlate with the symptoms that we use right now to diagnose mental disorders with. So Dr. Gray, thank you so much for a fantastic talk and for a stimulating discussion. That was terrific. There's some questions that I couldn't get to. I'm sorry, if you wouldn't mind staying on and answering them, that would be great. And for those of you that sent pre-submitted questions, we couldn't get to a lot of those, but we'd be really happy to follow up with you. And so sometime over the next few weeks, we hope to be able to send you answers to those. Okay, so that ends the section on the speaker from our two keynote talks. I want to thank both of them for terrific talks. And now I'm going to turn to the next section, which is the data blitz. And to do that, I want to mention and thank our NeuroFest co-chairs, Dr. Tim Hanks, who's working behind the scenes, and Dr. Kostas Zarbalis, who you're going to see next, who's going to run the data blitz section. Now, these two co-chairs, along with Jennifer Scott and our amazing students, have really worked to organize this event. And I really owe them a huge debt of gratitude. It's going incredibly well, and we want to thank all of you for all of your work. So as I mentioned, Dr. Zarbalis is going to lead this next section. And so I'm going to introduce him a little bit. He is an associate professor in the Department of Pathology and Laboratory Medicine at the UC Davis Medical Center. Dr. Zarbalis is also a member of the Institute for Pediatric Regenerative Medicine and the Shriners Hospitals for Children. His research focuses on forebrain development in health and in neurodevelopmental disorders. So Dr. Zarbalis, it's all yours.
Thank you very much for the introduction, Kim. And now we are moving to, for the first time, NeuroFest brings you a new and exciting competition called NeuroBlitz. So NeuroBlitz challenges our graduate students to sum up their research for the general audience in a concise yet informative way. Similar to an elevator pitch, each student will have 3 minutes to present their research. As a member of the audience, we need your help. Please vote for your favorite NeuroBlitz presentation based on how well the students engaged with the audience and how clearly they communicated the significance and key concepts of their research. Once all of our contestants have given their brief talks, we'll launch a poll on your screen to collect your vote. The students with the most votes will receive a $100 award. So the winner of today's presenters will be announced during the interactive booth around 12 noon. So, and now I'm pleased to introduce today's NeuroBlitz graduate student contestants, starting with Olga Vafeva, who joined the UC Davis Neuroscience Ph.D. program in 2018 and currently trains in the lab of Dr. Elva Diaz. She studies the generation of new neurons in the adult hippocampus and how this process changes with age. Olga, please.
Can an adult brain make new nerve cells? Well, if you were to ask Ramonica Hall, who is known as father of modern neuroscience, the answer is going to be no. So you might wonder, how come the title of my talk is studying new neurons in the aged brain then? Well, that is because recent discoveries have shown that there are indeed new neurons being generated in certain regions of the brain. For example, one of such regions is hippocampus, and that's what you can see here in red. What we've known for a long time is that this region is involved in learning and memory, as well as in mood regulation. However, what we learned just recently is that this brain region also continues to make new nerve cells throughout life in the process that's known as neurogenesis. And to understand neurogenesis, we need to look deeper. So if you were to slide through the hippocampus and look inside, that's what you're gonna see. So in blue, you can see labeled hippocampal cells. But if you zoom in even closer, you can see that some of these cells are also labeled in red. And these are very special cells. They're called neural stem cells. And their main functions is to make new neurons. And that's what they do throughout life, allowing neurogenesis to happen. And these new neurons have a variety of functions. For example, they're important for spatial navigation, helping you to move around the world. Also, in memory refinement, by allowing you to discern between similar yet distinct memories. But one of the most intriguing ones is its role in mood regulation, as it has been shown if you make fewer of these neurons, you end up with depression symptoms. So that's a pretty neat concept to have these new neurons being generated throughout life, contributing to important brain functions. However, there is a catch. You know, as you grow older, the rate with which these new cells being generated decreases, meaning as you age, you make fewer and fewer of these neurons, which possibly affect the functions that I mentioned before. So here at UC Davis, in the labs of Dr. Chang and Dr. Diaz, we're trying to understand what makes these cells so special and why there are so few of the neurons being generated later in life. But while we're looking for answers, there are some ways you can already reverse this process. For example, you can increase number of newly generated cells by just running. Well, if you're not into running, no judgment here, neither am I. There are foods that can help you make more cells. For example, antioxidant-rich blueberries, or my personal favorite, flavonoids that you can find in dark chocolate. So if you want to stay sharp, jog in the morning, enjoy blueberries and chocolate more often, and make new neurons. Thank you.
Thank you very much, Olga. That was beautiful. So we are moving now to the next neuroscience student. This is Katie Roning. She's a senior neuroscience PhD candidate in the lab of Dr. Mary Burns. Katie's research aims at improving our understanding of how immune cells act in the central nervous system in health and disease. Katie, please.
Let's start by zooming in the central nervous system to the cellular level. During normal, healthy neuronal function, there are a variety of cells that work together to keep everything running smoothly. We have neurons receiving and transmitting information rapidly, glia, which support neuronal function, and my personal favorite cells, the immune cells. During this normal neural function, these immune cells are constantly surveying their environment, keeping a sharp lookout for anything amiss, and working to help clean up the debris and waste that's just naturally generated by these hardworking neurons. However, sometimes things don't always run so smoothly, and unfortunately, in some cases, neurons begin to die. This is known as neurodegeneration. There are a range of neurodegenerative disorders you might have heard of, ranging from macular degeneration to Alzheimer's disease and more. And while there are important differences between these, at the core of each of these devastating disorders is this loss of neurons. There are many scientists working hard to understand why neurons are dying in these disorders, but I'm really interested in these immune cells. We've known for a long time that these immune cells can sense and respond to neuronal damage and death, but what are they actually doing? This is an important question because we know that throughout the body, immune cells can generally adopt two distinct functional states. Sometimes they can be helpful, which is like what we see during this normal neuronal function. But sometimes they can actually be harmful and cause further damage while they respond to the surrounding disease. So during my dissertation, I've been asking, during neurodegeneration, are these immune cells working to prevent neuronal death, or are they accidentally contributing to it? Using techniques that allow us to analyze individual cells in mouse models of neurodegeneration, myself and others have found it turns out the answer is both. Simultaneously, during neurodegeneration, some of these immune cells have a more helpful state, working to try to save the surviving neurons and prevent further neuronal loss. But others have this more harmful state, actually contributing to the loss of neurons. Although this is complicated, we're really excited to have this avenue to pursue new generations for therapeutics. Now that we have a better understanding of the varied roles these immune cells play during neuronal loss, we're shifting our attention to the signals that can push immune cells to either of these states. We're now asking, can we harness this information to develop therapeutics that can push immune cells away from this harmful state where they cause further damage to this helpful state so that we can use all of these immune cells to help us combat this loss of neurons? There is a lot more work to be done for sure, but we're really excited to use this as a springboard to try to create new interventions for neurodegenerative disorders.
Thank you very much, Katie. And this brings us to our last contestant. This is Tracy Warren, who joined the UC Davis Neuroscience PhD program in 2018 and trains in the lab of Dr. Alex Nord. She's very interested in understanding how the genomic regulation of biological pathways in the brain may lead to psychiatric disorders. The stage is yours, Tracy.
Hi. So I thought we could start off by talking a little about the structure of DNA. Your DNA contains genes. You all know that. But what you might not know is that the term genes actually only describes a small section of your DNA. They're what make the products, proteins, that your body uses, so we call them coding. But outside of your genes are enormous stretches of DNA that don't make anything at all. We call them non-coding. Rather, they contain instructions for how to read your different genes. So for example, genes that, liver genes only get read in your liver, and bone genes only get read in your bones. But something weird happens when we look where results from psychiatric DNA screens tend to be located. It's almost all outside of these protein coding regions. This is data from schizophrenia, but even for all disease as a whole, it still hovers around a 90-10 split. And I mean, that's pretty wild. So if these genomic changes aren't making messed up proteins, what are they doing? One hypothesis is they are enhancers, which is a kind of non-coding DNA that I study. Enhancers activate particular genes to be read by our body when they're important, all to create a healthy human organ. So let's imagine what happens if an enhancer gets broken. Well, then the gene can't get read like it's supposed to. And you wind up with this whole cascade of events that can create a big dysregulated brain. So how do we figure out if something is an enhancer? We have this cool tool where we can link up a potential enhancer to a gene that glows in the dark green when it turns on. We then inject this into mouse brain, and if the enhancer turns the gene on, the neurons literally glow green. And we know that the DNA we tested was in fact able to enhance that fluorescent gene. Our lab has used exactly this technique to test more than 400 regions of DNA that increase risk for psychiatric disorders. In this image, the cells that are glowing green received one of these enhancer green gene pairs, and the enhancer activated the gene to produce its fluorescent activity. So we think that some of the DNA that gets broken in these disorders do appear to be enhancers of their particular gene. Knowledge of what these mutated regions in psychiatric disorders are doing is amazing, because if we know what's going wrong, maybe we can fix it. And if we fix these broken enhancers, we might eventually be able to heal the brain itself. So I'd like to thank my amazing lab for their support and work in this project, and particularly shout out to our former grad student, Linda Sufer-Here, and our current postdoc, Jason Lambert, for their work in helping me push this project forward. And thank you all for your time.
Thank you, Tracy. And also to all the other NeuroBlitz graduate students, you all did a fantastic job. Now we'd like to invite our attendees to vote for your favorite NeuroBlitz. I'd like to ask Teresa to please launch the NeuroBlitz poll, which you will see on your screen momentarily. Simply select the name and topic of the NeuroBlitz you enjoyed the most and submit. The candidate who receives the most votes will receive a $100 award. I will look forward to announcing the winner of today's NeuroBlitz at approximately 12 noon. So while you cast your vote, I'd like to turn the screen over back to Dr. Kim McAllister.
Okay, terrific. So aren't our students amazing? The Data Blitz is just such a highlight of our new virtual format here at NeuroFest. So I really want to thank you, Kostas, and the NeuroBlitz contestants. That was really a lot of fun. While all of you, I hope, are casting your vote for your favorite NeuroBlitz, I want to share just a few final remarks. And the first is that I hope that you have planned and reserved time to spend the next hour or so visiting our interactive booths. We have a number of these booths that are outlined here. They are a lot of fun. And we've got activities for all ages, really from kids to the young at heart. And so I hope that you'll be able to attend these booths. The booths, as you should know from your invite, are only accessible using the online platform called Gather. Jennifer, if you could please add the access link to Gather into the chat, that would be very helpful. The platform looks like this. You navigate your avatar that you will have, that you can actually personalize and gather around using the arrow keys to each of these different rooms. And you just follow the arrow until you actually see the room. And then once you go into the box, you enter the room and you can interact with people. It's really, really quite a lot of fun. So the interactive booths are going to be open for the next 2 hours. So you've got plenty of time to explore them and to ask lots of questions and learn about the brain. So if you did not sign up to visit the interactive booths ahead of time, but you would like to now, there's no problem. That you want to visit the link here. It's the link that's actually in chat now, so you can click on it right from the chat from Zoom. You want to log in using the email address that you registered for the event for, so that's important. You may need to go through a one-time verification process. That's basically once you put your email address in, they're going to send you a verification email, open your email, click on that, and then you'll be able to enter Gather. There's a step-by-step set of instructions in the chat. And we're going to have a number of our administrative staff that are supporting this event remain in the Zoom room. So you can come back to Zoom using your Zoom link anytime to ask questions over the next 30 minutes. After that, we're going to close the Zoom room. And the winner of the NeuroBlitz will be announced in Gather. Okay, so Jennifer, if you could please add the link into chat for the step-by-step instructions, that would be really good. And just to remind everyone, once you want to head to Gather, you want to close Zoom. You don't need to stay in Zoom at all. And then once you're in Gather, you'll be good to go. So let's see. I want to end by, again, thanking the many people that made this program possible. Our NeuroFest event committee staff and volunteers have been working really hard, and you guys have done just a phenomenal job. So for all of the attendees, please join me in thanking the NeuroFest co-chairs, Dr. Tim Hanks and Dr. Kostas Zarbalis, the graduate student leads, Olga Vathayeva, Sasha Mikhailova, and Becca Wilson, the NeuroFest staff, Jennifer Scott, Teresa Gear, Kimberly Cummings, and Faye Vieres. You don't see them, but they've been making all of this go smoothly. Jennifer Scott in particular has also been involved in every aspect of this. So thank you so much, Jennifer. And to all of our graduate student volunteers and faculty who are involved in the interactive booth experience, it really is the highlight of the event, and I'm looking forward to seeing you all there. So with that, I'd like to thank you all for joining us. And we will see you next year in person, if we have anything to do with it, at NeuroFest 2022. Thank you, and please take care.